Hello everyone, welcome to Easy Biology. The topic is about transport of materials through plasma membrane. So already we know that cell is called basic unit of life. And it is also called structural and functional unit of living organisms. So some substances are entering into the cell and some substances are exit from the cell. Okay, for example, oxygen in or out. So oxygen is into the cell. Carbon dioxide from the cell to outside, so out of the cell. Glucose into the cell. Amino acids into the cell. Fats into the cell. Wastes out of the cell. Water into the cell or out of the cell, depending upon the need. Okay, next. So there are solutions which we have to prepare for understanding passage of materials through plasma membrane. One is the dilute solution. The solution in which water quantity is more. The solution in which water quantity is more is called dilute solution. Next one, concentrated solution. The amount of solute present in 100 ml water is called concentrated solution. Next, saturated solution. The solution in which solute is present at bottom is called saturated solution. Next, how solution is prepared? Because of the presence of combination of solute and solvent. Right, what is solute? The substance that is in small quantity. The substance that is in small quantity and dissolve in solvent is called solute. What about solvent? The substance that is in more quantity, and make solute to dissolve in it. It is called solvent. So solute and solvent forms solution. If the solute is salt, then the solution is salt solution. If the solute is sugar, then the solution is sugar solution. Okay, right. Next, one small activity. Here we have to take 3 beakers. In these 3 beakers, what we have to do is take equal amount of water. In 3 beakers, take equal amount of water. So this is the first beaker, second beaker, third beaker. To the first beaker, add half teaspoon sugar. To the second beaker, add 1 teaspoonful of sugar. And to the third beaker, add 1.5 teaspoon of sugar. Okay. Now the question is, can we convert first beaker into third beaker? And can we convert first beaker into second beaker? Okay. So we can convert first beaker into second beaker. How? By adding half teaspoon of sugar, we can change first beaker into second beaker position. Means nature. And by adding 1 teaspoonful of sugar, so that we can convert first beaker into third beaker. Okay, right. Now keep it aside. Now, can we convert third beaker into first beaker? Okay, and can we convert second beaker into first beaker? So it is possible, but it seems to be a little bit difficult. The reason is, to change these two into first beaker, what we have to do is, we have to add water. We have to add water, means a solvent. Okay, next. In this lesson, we are going to discuss about 2 physiological processes. One is the osmosis, another one is the diffusion. So first we will discuss about osmosis. Actually, osmos is taken from Greek word called osmos. Osmosis is the word which is taken from Greek word osmos, which means to push. To push. What is osmosis? The movement of solvent molecules from high concentration to low concentration is called osmosis. Okay, or we may also write like this. Whenever we taking solvent as criteria, the definition is like this: movement of solvent molecules from high concentration to low concentration. When we take solute word as the criteria, the definition is like this: the movement of solvent from less solute concentration to more solute concentration. The movement of solvent from less solute substance or concentration to less solute substance to more solute substance or concentrated substance. Now we will discuss about the osmosis process through activities. So what is the first activity is, we have to take a beaker. In this beaker we have to take tap water. Okay, Here what we have to place a dry resin. A dry resin in the sense or kismis. Okay, so this is the dry kismis in normal tap water. Now after leave it for few hours. After few hours we observe that the kismis becomes swollen. The kismis becomes swollen. Why? Because the water from the beaker enters into kismis. Next, now take here saturated solution. What is saturated solution? The solution in which still there is solute at the bottom, means where water is needed, that is called saturated solution. Here whatever the kismis swollen, place here. Okay, when we kept the kismis like this, it changed into this position because water is absorbed into the kismis from beaker, and whatever the kismis swollen we are placing in saturated solution. So this is the tap water, this is the saturated solution. Right, now what will happen? Whenever we leave for few hours, the kismis shrinks. The kismis shrinks because the water from the kismis enters into beaker. Why? Because to dissolve the solute present in the beaker, the water from the kismis exits from the kismis. The water exits from the kismis. Okay, so here who needed water? Beaker needed water. To dissolve the solute, beaker needed the water. Here the solvent concentration is more in kismis, whereas in this activity the solvent concentration is more in beaker. That is why water move from high concentration to low concentration, means water from beaker to where not there is kismis, so water enters into kismis. But here when we place the swollen kismis in beaker, water from swollen kismis enters into beaker which contains saturated solution because this beaker now required water. Okay, right, next activity. So before discussing about another activity, we will write here two more definitions, that is the movement of solvent molecules into cell is called, the movement of solvent molecules into cell is called end osmosis, whereas the movement of solvent molecules from cell to outside, means exits from cell to outside, is called exosmosis. So X means exit. If the solvent molecules enter into the cell, it is called end osmosis. If they are sending from inside to outside, it is called exosmosis. Next, activity with a potato cup. Second activity. What is this activity? Is for this we have to need a potato, and we have to cut the potato in the shape such that cup shape, potato cup, and take a petri dish. In this petri dish, we have to take normal water. In this petri dish, we have to take normal water. Right. Now, whatever the potato cup is there, okay, in this potato cup, take saturated solution, either salt saturated solution or saturated sugar solution, right, like this. Okay, right. So in this what we taken is, so this is the normal tap water, this is the saturated solution, either sugar or salt, right. Now what we have to do is take a pin and pierce it into the potato cup. Take a pin and pierce it into the potato cup and mark the level of water up to where the water level is there. Just mark the water level and leave it for few hours. Actually we know that who needed water here, right? Saturated solution needed water. Why? Because whatever the solute present in saturated solution, it has to dissolve. That is why to dissolve this saturated solution, water from petri dish enters into potato cup through this membrane, through the membrane. As a result, what will happen? We observed that the water level crossed the mark. The water level crossed the mark. It will come some more water level is some more raised. Okay, it is because the water from the petri dish enters into the potato cup. Okay, right. Now next, now what we will do is we have to take in petri dish saturated water or saturated solution. Saturated solution. And in potato cup we have to take normal tap water, right. Now what will happen? Again same, we have to pierce a needle into the potato cup and mark the level. For example, the water level is up to here is there, for example. So all this is the water, okay, and the water level is here is there, water level up to here is there, okay. So we know already saturated solution required water. When we leave the entire setup for few hours, we observe that the water level decreased and it crossed down the mark, whatever the indication we mentioned in the pin. So the water level comes down below this area, comes down. It means what? The water from potato cup enters into petri dish, right? So here potato, whatever the membrane is there, it acts as a permeable membrane. Here we have to know some properties of the membrane. For example, if the membrane allows substances, if the membrane allows substances to pass through it, if the membrane allows substances to pass through it, is called, this property is called permeability. If the membrane does not allow, if the membrane does not allow substances to pass through it, is impermeable. So if the membranes are allowed through the membrane, if the substances are allowed through the membrane, it is called permeability. If they are not allowed, it is called impermeability. Next, if the membrane allows solvent but not solute, then the membrane is said to be, then it is called semi-permeable. What is semi-permeable membrane? Is the membrane allows solvent but not solute. If The membrane allows solvent and some selected solutes. Then the membrane is said to be have selective permeability. Then the membrane is said to have selective permeability. Okay, right. Next, so actually plasma membrane is a selectively permeable membrane which is a delicate and thin layer surrounding the animal cell, and it is also present in plant cell below the cell wall. And one more activity to explain the osmosis. What is that is, for this we have to take egg membrane. So first of all we have to boil the egg. After removing shell, we observe there is a thin layer on the surrounding albumin. That layer, without breaking, we have to collect. After collecting it to that membrane, into this, with the help of syringe needle, take saturated solution. Now place this membrane in beaker containing tap water, and left with a small holding with the help of thread. After a few hours, we observe that water move from beaker to egg membrane. Water move from beaker to into the egg membrane. So here egg membrane acts as a semi-permeable membrane. So we can also prepare a semi-permeable membrane, and we can also perform reverse activity. Means here we are taking normal tap water. Here we are taking saturated solution, means in beaker, and this is the normal tap water. Now what will happen is, the water move from egg membrane to beaker, because beaker is containing saturated solution. So water move from egg membrane to beaker. Okay, right. So where we are using the osmosis in our daily life? So for example, liquids are moving in our body through osmosis only. Okay, between cells in our body, which process occurs is osmosis. Osmosis occurs. Okay, next, plants observe water through roots by osmosis, and reverse osmosis is the process through which salt is extracted from seawater. Salt is extracted from seawater or sea. Okay, so there are many functions of plasma membrane. What are the different functions performed by plasma membrane is, we will write. Functions of plasma membrane or cell membrane. So it gives definite shape to cell. It acts as a recognition center for another cell to identify it. It performs endocytosis, means to kill harmful antigens or pathogens trying to enter into the cell, and it maintains cell continuity. cell continuity between other cells, and it helps in absorption by modifying. Means, for example, villi in small intestine, villi in small intestine, or microvilli in small intestine perform so osmosis process, and these are the functions of plasma membrane. And we will discuss about another topic, diffusion, in the next class.